In this video, we're going to compare the gas exchange surfaces in different respiratory systems. We'll start off with the one that we know the best. So let's have a think about how mammals, in particular humans, exchange gases. So we have a very complex series of tubes leading from the outside world, going down the windpipe, also known as the trachea, which then branches into the left and right lungs, using the bronchi, or the bronchus would be one, and then that tube branches into smaller tubes called bronchioles, and they culminate in alveoli, which are little ball-like structures, very small, kind of like clusters of grapes, and each cluster is surrounded by an extensive network of pulmonary capillaries. So they allow the gas exchange to be incredibly efficient because the blood supply is running really close to the external air. We have to think about the features of an efficient gas exchange surface. So we're thinking about how to promote diffusion from the air to our blood and from our blood to the air. And there are a number of features that appear in all gas exchange surfaces. So we've already seen that there are lots of alveoli at the end of each of those branching bronchioles, which means there's a very large surface area. In fact, inside a human, an adult human, it's probably about 70 square meters of surface area inside our lungs. In this image, you can see that in order to promote efficient gas exchange, we also need great ventilation. In other words, we breathe deeply and keep mixing old air in our lungs with fresh air from outside. So we don't completely replace all the air in our lungs, we just mix some fresh air in with the old air, just to keep the concentration of oxygen high and keep removing the CO2 that we've put into the air inside our lungs. So that's good ventilation, another aspect of an efficient gas exchange surface. The next one is moisture. So all gas exchange surfaces are moist so that the gases can dissolve into that moisture layer and get very close to the gas exchange surface. The inside of our lungs is moist. We know that when we breathe out, you can feel moisture on your exhaled air. We also have a very short diffusion distance. So if you have a look here, you can see that there is one cell that's lining the alveoli. So that's the edge of our alveoli is actually only one cell thick, and the edge of this capillary here is also one cell thick. So in order to diffuse gases from the outside air into our blood, we actually only have to go through two cells. So 2 cells thick. That's a very short distance. It's in the micrometers distance, so that's going to promote efficient gas exchange. The next thing is good circulation. So you can see this capillary here, incoming on the left, we have deoxygenated blood, so it'll be rich in carbon dioxide and depleted in oxygen. It's coming in from the heart along the pulmonary artery, which has branched and branched and branched to form these capillaries. And each of these capillaries runs really close to the alveoli, and as it leaves, it takes away the oxygen that has gone into the blood and is ready to pick up some more CO2. So this is helping to maintain a great concentration gradient, which is our next point for having an efficient gas exchange surface. So we want to make sure that we're always bringing blood that's rich in CO2 to the alveoli, removing that, and then taking away the oxygen. So we want air that's rich in O2 to move that oxygen in. So we're always thinking about moving gas from where it's in a high concentration to where it's in a low concentration, so down a concentration gradient, because all of this movement of gases is passive. It's just happening by diffusion. So those are the features of a good gas exchange, and they're all summarized on this one diagram here. But let's compare this to other gas exchange surfaces in other creatures. So in insects, we have a large surface area because the system of tubes that allows oxygen and sort of air in and out of an insect, so oxygen is going to be picked up from the air and CO2 is going to be removed, happens through a series of tubes. They open to the outside world through these little holes called spiracles that are connected to tubes called tracheal tubes. These branch and branch throughout the insect, and they end up in really little tubes called tracheoles, which come very close to every cell of the insect's body. And you can see that at the end of these tubes, the gases are diffusing out, if it's oxygen, or back in if it's CO2. And they're little valves at the openings of these tubes to the outside air, so that some tubes allow the air to move in as the insect kind of pumps its body, and other tubes let the air out. Not all insects pump their body to ventilate their body, but the bigger ones tend to. So big bumblebees or a big grasshopper like this one. So they have a large surface area, good ventilation with the pumping and the tubes. They're also moist because there's moisture at the opening of these spiracles that will layer the inside of the tubes. There's a very short diffusion distance because of the massive branching that these tubes do and the closeness they get to all the cells. There is no circulation involved in an insect's oxygen transport because they don't use their blood system for carrying oxygen around. However, they are maintaining a concentration gradient because they're always moving that gas around their body. The next animal we'll look at is a fish. Fish use gills as their gas exchange surface. And every gill has little filaments on it, which gives them a huge surface area. They're ventilated because water moves in through the fish's mouth, and then the fish will close its mouth and push the water out over the gills, so they get well ventilated with fresh water the whole time. Obviously, they're moist. The fish lives in water. And the gill filaments are very fine, and they are actually protected by an operculum or a gill cover to keep them safe from any damage or any parasites or any attack. A nice hard cover because they are very delicate, because they're literally one cell thick, just like the alveoli in our lungs. You can see they're bright pink. That means there's good blood circulation, and the concentration gradient is maintained because the direction of water flow is opposite to the direction of flow of blood in the gill filaments. So you always get a good difference between the concentration of gases in the water compared to the concentration of gases in the blood. And here is our final creature, a frog. Now, a frog's inside lungs aren't particularly well developed. They're not like a human's lungs, but the frog has an advantage because it also can gas exchange through its skin and in the lining of its mouth. So there are more surfaces that it can use. And often its skin is quite sort of folded backwards and forwards as well, and quite wobbly, like you can see on this frog. So there is actually quite a large surface area for the size of the frog. You can see ventilation. The frog breathes, pulling air in and pushing it through from his throat into the lungs. You see the lungs kind of expanding. Inside, you've got delicate tissue, just like we have in our alveoli. So it's a very short distance for that oxygen to diffuse into the blood and for carbon dioxide to leave. And the frog has a heart pumping blood around its body. So the concentration gradient is maintained with the flow of blood and the ventilation in the lungs, just like we do. So in summary, that's comparing all the 4 different creatures that we've looked at here in terms of the organs that they use to gas exchange, how they keep them moist, how they keep the surface area big, and how they transport the gases to and from the respiratory surface.